Hi, so my name is Fiona McCartney and I work in University College Dublin. So a bit about my background. I am actually from Ireland and I'm from a place called Dundalk, which is near, actually on the border between Ireland and Northern Ireland. And I did my degree there in biopharmaceutical science. And then after that, I went on to University College Dublin to work in Professor David Braden's group. We're actually based in the UCD Veterinary Medicine School. So our group mostly looks at drug delivery, in particular oral drug delivery, but we're also looking at intra-articular and buccal delivery. So kind of during the whole process of Transint, I have moved from being a PhD to a postdoc. And my actual PhD thesis was focused mainly on oral drug delivery, with a particular emphasis on intestinal permeation enhancers and nanoparticles. And kind of, especially with the intestinal permeation enhancers, I kind of moved from the in vitro, ex vivo to in vivo situation. So I used CACO2 cells grown on transwells, and then I also used and spent most of my time with this particular piece of equipment, which is also an Ussing chamber, a bit like what Patrick has. And we're able to use tissue that we're able to mount in this and look at fluxes from the apical to basolateral side, or the other way if that's what we choose. So sort of my role within Transint, I suppose, was particularly focused on the question of oral insulin and is that possible? And particularly my role was related to testing nanoparticles for their in vivo. So as you can see, sort of nanoparticles, the question is, if they're delivered orally, they get into the gut, are they able to then either themselves get out into the bloodstream or release the peptide and allow it to get out into the bloodstream? So my particular role involved a number of people from throughout Europe and throughout the Transint project, different universities sending me their different nanoparticle prototypes, and then I was able to test them within our in vivo model. So as you probably saw earlier in Professor Braden's talk, we particularly focus on the intestinal loop method. We've a history of using that with intestinal permeation enhancers, and this is the first time we've actually used it with nanoparticles. So for this model, it's an anesthetized animal, it's non-diabetic, and what I do basically is I create this kind of loop. So I tie off this end, and I'm able to inject the nanoparticle solution into the loop, and then I'm able to put it back in, and then I'm able to take samples from the animal and basically able to see if we see an increase in serum insulin or a decrease in the blood glucose levels. One of the things we're able to do after this is actually preserve the actual loop itself. So then I preserve that, and then I'm able to do microscopy, so we can either look at basic morphology with the H&E staining, or look at mucus production with Alcium blue, or in some cases the partners were able to send me fluorescently labeled or tagged nanoparticles, and for those experiments I just instilled them into the loop, waited 2 hours, and then at the end of the experiment I was able to take the loop out, and then I was able to look at the staining of the tissue. So as you can see, we have your villi here, and you see your fluorescent nanoparticles just here. And then I was also able to quantify the insulin in the blood. I was able to use ELISA, and also in some cases it was necessary to send it for LCMS testing. So this is kind of representative data. I'm just calling it nanocarrier and not naming it in this case. So we just kind of have a PK and a PD readout for this. So in this case, it's our pharmacodynamics. So I look at decreases in blood glucose levels over time. So I have a PBS control, an insulin solution control, which is at 50 IUs per kilogram, which is the same dose that the nanoparticles are at. And I also, it's not shown here, but for each case of nanocarrier that I tested, I also tested an unloaded version. Or in the actual case of this nanocarrier, it was not possible to form the nanocarrier without the insulin being present. So I tested the excipients that were used without the insulin present. So as you can see, for PBS and insulin solution, I don't really see a decrease in the blood glucose levels, where I see a significant decrease over the 2 hours for the nanocarrier. And then again, we have our PK readout, and again, no real effect of seeing insulin in the serum, whereas with the nanocarrier, I actually see an increase over the 2 hours. So if we look at the actual readouts from the PK, as I said, I also did, as a control, and for comparison, I did subcutaneous injection of 1 IU per kilogram of the insulin. So as you can see, the Tmaxs are slightly different. So you get 20 minutes for the injected insulin, and then for the nanocarrier, it was about 53 minutes. And then the relative bioavailability I was able to achieve for this nanocarrier was 3.8%, and the pharmacological availability, which is the PD parameter, was 2.5%. So what have I learned? And that's probably the biggest thing. There's almost, as Matilda was talking about, there's two parts to being a PhD during Transient, is that you have your scientific learning. So I learned all about nanoparticles, which is not an area I was particularly familiar with. I got to interact with a lot of people who work in this area, but also the idea of being sent a formulation that involves a lot of logistics and organizing and discussing with the different people how you're going to do that, what dose you need, how much solution, do you need this or whatever. Also, the most important thing is that these kind of projects need collaborations, that it's not just one group able to do it on their own. We're able to work together with different pieces of the puzzle. And also, the biggest learning I found was that it's really, really important for the people who are formulating the nanoparticles actually to talk to the people who are doing the in vivo studies, because it's all well and good to formulate your nanoparticle, but then realistically, can you actually provide the dose that they want of, say, 50 IUs per kilogram? Or what are you going to have to do? You may need to concentrate your nanoparticle to do this. Also, and one of the issues I came across was some particular formulations were viscous, or so it's actually, a lot of people test and do a lot of their physical chemical testing at one concentration, but that may not necessarily be relevant to the in vivo situation. So the nanoparticle or nanocarrier itself can change based on the dose that you're needing, and that can affect its physical chemical properties. So one of the other great things about Transient, and I think it's particularly important for our students to have, which is an industry partnership. So during Transient, we were able to, I worked particularly with Sanofi, and then also with an Irish company, Sigmoid Pharma. And then also as part of Transient, Patrick, who gave the last talk, came for a day to our lab in Dublin to learn a particular technique. And then also I collaborated with IRB in Barcelona on looking at cell penetrating peptides. And Joseph here came to visit us for 3 months. So it was really good to have this exchange of ideas. And then one of the things that you really get an experience of during Transient was presenting. So we normally have yearly meetings, we also have teleconferences, and it's really good experience for PhDs or postdocs to have to present to quite a mixed audience, which would include industry partners, EU advisors, and actually be questioned on your work, which might be scary at the start, but actually it kind of builds this ability in you to not be scared by getting asked questions, but also it makes you think about things in different ways. And so then finally, my outputs. So I had oral presentation at the Irish Epithelial Biology Society. I had an oral presentation at the conference that Transient was involved in organising in Dresden, which is Crossing Biological Barriers. Also a review on intestinal permeation enhancers and their safety. And then our group also created a YouTube video about what we do in our lab, in particular reference to Transient. I had, within UCD, we have a Conway Festival, and 2 years in a row I had moderated poster, and last year I also had oral presentation. In terms of CRS, or Controlled Release Society annual meeting, at the Edinburgh meeting in 2015, I think it was, I had, I think it was 5 abstracts, 3 of which I was lead author on, and 1 of which was with IRB in Barcelona. And this year at the Boston meeting, I have oral presentation in the biologically active excipient category. So, thank you. Great. Oh, sorry, what next? Nearly forgot. So yes, as I said, I've gone from a PhD to a postdoc. So it's kind of done the whole journey. Now I am a postdoc, partially working with Transient, which will end now. And also I'm working with GAFOSA, so they fund my postdoc, and so I'm looking at different lipid-based excipients, which is a change, and also I'm doing a tiny bit of teaching to get experience. Okay, finally finished. Thank you.
So Fiona will graduate in June with her PhD conferring.
Yes.
Okay, any questions? Time for one maybe? Go ahead.
Okay, I suppose in a way we had experience with non-diabetic animals and with our particular intestinal insulation technique, but one of the good things about insulin is you will get a response whether the animal is diabetic or not. So we see it almost as a pre-screening tool. So it's a way to kind of quite quickly work through a series of nanoparticles, and something has been brought up as well is something like the STZ model can show a lot of variability, and it's quite a harsh model. So this was just more a way to kind of, as I said, do a kind of pre-screen and look at these nanoparticles and see if they actually did have an effect. And one of the good things about this is in the same animal I do PK and PD, so I'm able to correlate them together and kind of see not only are they having a response, but how much of the insulin is actually getting into the bloodstream.
Okay, very good. We're going to move on.
Okay.
One more. That was last one.
So I tested some from here in Santiago. So basically what other people have presented on today, I've tested some of those. So I tested, that one is actually the one I can't. Yeah, it's one of our particles I can't talk about. Okay, so what I ended up doing was everyone sent me a solution. So if they made their nanoparticle up in water, that's the way it was injected. I literally asked everyone to give me a concentration that I could calculate approximately. It varied depending on, and this is where I came into it, whereas for some people they were able to make a certain mgs per mil of insulin, which meant I only had to give, say, 250 microliters, but for some people that was not possible and I had to give a higher volume. So what happened was they basically sent me their solution or suspension, told me the mgs per mil of insulin that, and I directly injected that in, so I didn't dilute any of the samples.
Great.
They sent me them water. There is some studies I did where I actually administered PBS at the same time to see if that would have an effect.